These are organoids, microscopic tissues made up of cancer cells that mimic how tumours grow inside the human body. This new way of growing cells allows researchers to study cancer more accurately than ever before. And a new global research project to develop more of these miniature tumours could help test the next generation of cancer drugs. For over 50 years, researchers have been growing cancer cells in the lab on flat plastic dishes.
On this occasion, Dr. Stoker is examining cultures of breast tumour cells from patients at the Guy's Hospital unit. The ultimate intention being to determine the sensitivity of a particular patient's tumour to treatment with drugs or hormones.
But studying cancer cells in this way has some drawbacks.
They no longer look like tumours or tissues that they've come from, that we know, that tumours generate in large sort of bulk masses within the body. And we also found that generally cell lines that grow in plastic have come from the most aggressive cancers, so our whole collection is completely biased.
Now, instead of growing only the most aggressive tumour cells, a pilot project led by Cancer Research UK and scientists at the Wellcome Trust Sanger Institute aims to develop organoids from esophageal, bowel and pancreatic cancers across all stages of these diseases. To do this, the researchers extract the cancer cells from a sample of a patient's tumour and grow them in a special 3D scaffold.
They don't look like a full colon or pancreas, but certainly at the microscopic level, at the cellular level, they are much more reflective of the biology of the tissue from which they came. So in my lab, we're trying to understand why some cancers are sensitive to a drug while others are not. And so what we do is we test hundreds of cancer drugs against cancer organoids growing in the laboratory. And so what you can see here is the way that an organoid has responded to a drug. And you can see what this shows is that this drug is killing the organoid in the laboratory.
Alongside this, the team are studying the faults in the organoid's DNA, creating a genetic passport that will tell scientists how each organoid responds to the different drugs.
And that means in the future, when we see a patient with the same genetic changes in their DNA, we'll know which drugs will work and which ones won't.